Hi, welcome to ESI 412, Nanotechnology, Materials, Infrastructure, and Safety. I'm Professor Ukjun Nam at the Pennsylvania State University. So this is the outline. We will talk about what is the molecular self-assembly, and then we will talk about the examples, and then we will talk about the forces and then ligands used for the molecular self-assembly. And then this is the kind of a special type of the self-assembly. We will talk about the DNA origami as well. So what is molecular self-assembly? Let's see the examples we are using actually this kind of technologies for the commercial production. So this is the pregnant test kit. This is the kind of commercial version. If we are just looking around the details about this test kit, it has an absorber tips, and then there is a nanoparticle reservoir, and then there is a filter. So in the nanoparticle reservoir, there are two types of nanoparticles present. When urines kind of applied to the absorbent tips, when those kind of hormones for the only available for the pregnant women is kind of available, all these kind of two different size of nanoparticles kind of bind together by the hormones, which is yellow in these cartoons. Upon these kind of reactions, the entire size of the nanoparticles are very big. In this case of that, these big particles cannot pass through the filters. So we will see some lines in this area. If these hormones doesn't present in the urine, these two different types of the nanoparticles never bind together. So those kind of two particles just pass through the filters and then just collected in the reservoir. So how we put this kind of biocompatible molecules to the nanoparticle, that's the techniques we're going to talk about today, the molecular self-assembly. So these kind of same techniques used for another kind of application. Using that kind of self-assembly, molecular self-assembly techniques, we can just put drug molecules on the nanoparticles and then put those kind of nanoparticles through the bloodstream and then just use that nanoparticles for the drug delivery. And this is another kind of example we are using in our daily life. And this is the one of the example of the self-assembly as well. So
latte or olay, cozy or cool, or exactly the way you want it until boom, it's bedtime.
So as we watched these videos, actually the soap uses molecules which has amphiphilic structures. So one end is on hydrophilic, and then the other one is on hydrophobic. So this kind of hydrophobic end of the molecules kind of combined with the dirt, and then as we just wash all these detergent in the water, those kind of hydrophilic molecules are washed away in the water. So using this kind of two different kind of characteristics of those kind of molecules, we can just wash using the soap. So what is the molecular self-assembly? Actually, there is two assembly. One is a static assembly, and then the other one is a dynamic assembly. So the static assembly uses some kind of thermodynamic free energies. So upon those kind of molecules assembled together, those kind of free energies kind of getting the minimum point. Dynamic assembly is kind of mostly depends on the kinetical reaction, so it does not necessarily fulfill those kind of thermodynamic energy minimum. So I will show you those kind of examples in the later slide. So sometimes this molecular self-assembly, we call that as an self-organization, same meaning. And then the definition is not that is not an not making some structures kind of creating for this kind of self-assembly. So in another word, the self-assembly is not the formation process. And then this one does not need any kind of very expensive tools to make this kind of reaction happening. And then which can make an approaches about very small sizes. So in our kind of cases to making those kind of nanostructures, we already have the design in our software. So we make the designs, created the design using the software. But in this case of that, we make those kind of designs on the materials. For example, we will talk about this in the next lectures. So, for example, block of polymer, we can engineer the material. So, using those kind of specific materials, we can just generating some specific kind of patterns. So, in this kind of self-assembly, we do not have a specific kind of a design. We created using the software as we used for the e-beam lithography or those kind of lithography techniques. So protein is the one of the example of the self-assembly. So there are three kind of structures of the hierarchy. So this kind of amino acid kind of just positioned each other and then just forming this kind of rope-like structures. And then these kind of rope-like structures combined with secondary structures and then folding these first and then secondary structures, forming a specific shape of the proteins. And then based on these kind of structures, those kind of function of the proteins are very different. Another example is a lipid bilayer, like on soap. This kind of a lipid bilayer, the molecules made out of this kind of a polar head group and then non-polar head tail. So those kind of depends on the polarities. These kind of molecules aligned with the same polarity, and then it's just making this kind of a one layer, and then here's another layer, and then just making this kind of a bi-layer. And soap is the another kind of example. We just watch the video. And then the block of polymer depends on the portion of the length of the material A and B. and then the molecular weight, that's the length of the molecules, we can just change the shape of these kind of patterns, and then we will go through about this more in details in the next lecture. So here is the forces and then ligand for molecular self-assembly, and then the forces are chemical bonding, and then other forces like an electrostatic or magnetic forces, and then like an lipid bilayer, those kind of polarity and shape, and then template, and those kind of things can be some kind of a force to making this kind of a molecular self-assembly process. So this kind of a molecular self-assembly process can be used to modify the surface of the materials, and then we discussed about that in the previous lecture for the nano imprinting. So using this kind of a molecular self-assembly, we can change the surface energy of master mold material, like from hydrophilic to hydrophobic. And then we also discussed about this kind of reactions using the trichlorosilane and then OH bond in the previous lecture. So as this kind of ligand materials sees the surface, it just coming down. Actually, those kind of ligand material has an two end. So one functional group can bind one end, and then the other functional group can bind to the other end. So we can put one end for the ligand materials, which can selectively bind with certain materials like gold or oxide. And then this kind of lengths of the polymers can be used as a spacer, which just controlling the distance or overall film thickness of this kind of self-assembled monolayer. And then depends on the application, we can put another kind of a functional group on the other end, the spot for the B. So if we wanted to make an some kind of a drug for the tumor, we can put those kind of a tumor, those kind of medicines or drugs, molecules into this kind of a B site, and then in this case of that, we can just make one nano particles to be a drug delivery application. So when this kind of molecules sees the surface, first it just chemisoft to the surface, and then by the intermolecular reaction like Van der Waals force, those kind of bind molecules just aligned toward to one direction, and then by those kind of two reactions, it can just compactly forming one monolayers like this. And then, as we talked in the previous lecture, depends on the size of the molecules. Those kind of reactions to forming this kind of monolayer is kind of aggressive or less aggressive. So the heavier the molecule size, those kind of reaction is kind of less reactive, and then the smaller the molecule size, the reaction is kind of much more aggressive. So this is the kind of a table summarizing those kind of ligand materials versus the surface. So this kind of a surface like an chrome, nickel, iron, and aluminum, we can use this kind of a ligand material. And then for the gold, we can use thiol or this kind of a sulfide, or there is a depends on the surface. This could be nanoparticles or some of the thin films. So depends on those kind of materials we want to modify. We can use different kind of molecules as a ligand. And this is another way we can forming this kind of organic film. So when we just put those kind of molecules on the water, those kind of molecules kind of floated on the water, and then using this kind of a two spacer, as we just make these kind of films much more condensed, we can just make these molecules aligned compactly like this. So in this kind of situation, depends on the hydrophobic or hydrophilic characteristics of the molecules. It has one end, and then the other end has a different kind of characteristics. So after we just making this kind of organic films to be dense, we put the substrate pull out the substrate from the reservoir. As we just pull out the substrate, we can just pull these layers together with the substrate, and then we can just forming these layers on a substrate. And as we just put this substrate, one layer called this substrate, back to the same reservoir, in this case of that, this kind of hydrophobic end just bind together, and then in this case of that, you can just making this kind of a bilayer formation. And using this kind of this is an alternative way. So using this way, we can just directly form hydrophobic molecules touching the substrate directly. So this is the opposite case with this. So using this kind of a way, we can just make this combination of the layers, or this kind of a combination of the layers, and then they are all depends on how we're gonna use this kind of a layers, and then we call this kind of layers formed by this technique as a Langmuir-Blodgett film. This is another kind of example using the kinetically self-assembled nanoparticles. So let's say there is a substrate, and then we just put a drop of nanoparticle solutions, which solvent is toluene. If we just leave this drop on the substrate, those kind of toluenes will evaporate, and as those kind of a medium just getting evaporated, those kind of nanoparticles just assembled on the surface of this kind of a drop, and eventually as this kind of solutions all evaporated, it's just forming this kind of a well-aligned nanoparticle layer. So in this case of that, we can align, and then we can deposit one monolayer of this kind of a nanosphere layer using this kind of a kinetical force. Another thing is using this kind of a different kind of a shape, we can just make this kind of a sample structures. After we just dice this kind of sensor structures, just making the shape of the substrate just match with this shape of the sensors, we can just make this kind of a small piece of the samples well aligned to a specific substrate. And in this case of that, self-assembly also used kinetic force. And then this is another kind of example, lock and key binding. So this is most of the cases when the drug molecules kind of bind with some biomolecules. So some small molecules just lock in the shape of the biomolecules. Using that way, those kind of drug is working. So let's talk about the DNA origami. So DNA is by this point, those kind of self-assembled monolayer molecule has only two functional groups in each of the end of the rope-like structures. DNA, we have four functional groups. So this ANT and then GNC, they specifically bind in this kind of a pair. So using this kind of a way, we can engineering further more complex kind of structures using this kind of a DNA. So let's see how this kind of DNA is kind of replicated and then synthesized. So this video shows the better understandings.
Did you know that cells in your body have to regenerate and replenish themselves? Well, think about what happens to your skin in the winter. Your skin becomes dry and flakes off. What happens to those skin cells? Does your skin become so thin from loss of cells that you can see through it? Of course not. The cells regenerate to replace the ones that flaked off. This occurs through a process called mitosis. During mitosis, one cell divides to produce an identical copy of itself. If the cell is going to be identical, it must also have the same copy of DNA. DNA replication is the process where DNA makes an exact copy of itself for a new daughter cell just before mitosis. DNA replication is semi-conservative. This means that instead of having an entirely new copy of DNA for the new cell, a parent DNA strand splits in half, and each half adds a new complementary strand. The result is that each of the two double-stranded molecules of DNA are half old and half new. Let's explore how this happens. The first step in eukaryotic replication is for the enzyme helicase to unzip the double-stranded DNA molecule. The enzyme literally separates the two strands by breaking the hydrogen bonds between A and T and between G and C. Once helicase unzips the two strands of DNA, there are single-stranded binding proteins that hold the strands apart. An enzyme called primase can then put an RNA primer on the DNA molecule. The primer provides a free 3' OH so that a phosphodiester bond can be formed with the next nucleotide. DNA polymerase binds to and extends the sequence from the primer by adding complementary nucleotides. How does polymerase know which nucleotide to add? Well, if the original DNA molecule has an A, then polymerase will add a T. What do you think polymerase will add if the original molecule has a G? That's right, it'll add a C. It continues along the DNA molecule, adding the complementary nucleotide. Remember that a double-stranded DNA molecule is anti-parallel. This means if one strand is in the 5' to 3' direction, the complementary strand must be positioned in the 3' to 5' direction. Newly synthesized strands of DNA must be anti-parallel to the parent strand they are being copied from. However, the thing about DNA polymerase is that it only synthesizes new strands of DNA in one direction, only in the 5' to 3' direction. Because polymerase can only extend in the 5' to 3 direction, one strand follows along behind helicase and can extend continuously. This is called the leading strand. The other side has to wait for helicase to unzip another section of DNA so that it can lay down a primer and polymerase can extend. Because polymerase must wait for the replication fork to open, this strand is called the lagging strand and results in discontinuous fragments called Okazaki fragments, named after the scientist who discovered them. Last but not least, an enzyme called ligase comes along and seals the nicks between the Okazaki fragments to ensure there are no gaps in the phosphodiester backbone. DNA replication results in two identical copies of DNA that are half old and half new. The strands are complementary to one another. This new copy of DNA is ready to be placed in a new cell.
So this kind of replication process is kind of
Biotechnology capitalizes on the attributes of cells.
so this replication process is kind of pretty useful when people just multiply those kind of DNA samples. So when people has DNA, and then if people wanted to multiply the number of the DNAs, people use polymerase chain reaction. So as people just increase the temperature, those kind of double stranded DNAs unzipped, and then as people just lowering the temperature down, they just it just making another kind of a strand. So just repeating this, it just keep multiplying the number of the DNAs. That's the kind of polymerase chain reactions, and then that's the kind of a pretty important reactions are used for another kind of an application. So the DNA to introduce to the more details about the DNA origami, we're gonna watch another video, which is a pretty good videos. And then actually the shape you are using you are seeing right now, these are all kind of a drone design, and then the images on this kind of 3 rows, they are all actual images based on the kind of made by the DNA. So we will see how these kind of structures can be created by the DNA.
So people argue vigorously about the definition of life. They ask if it should have reproduction in it, or metabolism, or evolution. And I don't know the answer to that, so I'm not going to tell you. I will say that life involves computation. So this is a computer program. Booted up in a cell, the program would execute, and it could result in this person. Or with a small change, it could result in this person, or another small change, this person, or with a larger change, this dog, or this tree, or this whale. So now if you take this metaphor as genome as programs seriously, you have to consider that Chris Anderson is a computer fabricated artifact, as is Jim Watson, Craig Venter, as are all of us. And in convincing yourself that this metaphor is true, there are lots of similarities between genetic programs and computer programs that could help convince you. But one to me that's most compelling is a peculiar sensitivity to small changes that can make large changes in biological development. The output, a small mutation can take a 2-winged fly and make it a 4-winged fly. Or it could take a fly and put legs where its antenna should be, or if you're familiar with The Princess Bride, it can create a 6-fingered man. Now, a hallmark of computer programs is just this kind of sensitivity to small changes. If your bank account's $1 and you flip a single bit, you can end up with $1,000. So these small changes are things that I think that indicate to us that a complicated computation in development is underlying these amplified large changes. So now, all of this indicates that there are molecular programs underlying biology, and it shows the power of molecular programs, biology does. And what I want to do is write molecular programs, potentially to build technology. And there are a lot of people doing this, a lot of synthetic biologists doing this, like Craig Venter, and they concentrate on using cells. They're cell-oriented. So my friends, molecular programmers, and I have a sort of biomolecule-centric approach. We're interested in using DNA, RNA, and protein and building new languages for building things from the bottom up, using biomolecules potentially having nothing to do with biology. So these are all the machines in a cell. There's a camera, there's the solar panels of the cell, some switches that turn your genes on and off, the girders of the cell, motors that move your muscles. My little group of molecular programmers are trying to refashion all of these parts from DNA. We're not DNA zealots, but DNA is the cheapest, easiest to understand, and easy to program material to do this. And as other things become easier to use, maybe protein will work with those. If we succeed, what will molecular programming look like? You're going to sit in front of your computer, you're going to design something like a cell phone, and in a high-level language, you'll describe that cell phone. Then you're going to have a compiler that's going to take that description, and it's going to turn it into actual molecules that can be sent to a synthesizer, and that synthesizer will pack those molecules into a seed. And what happens if you water and feed that seed appropriately is that it'll do a developmental computation, a molecular computation, and it'll build an electronic computer. And if I haven't revealed my prejudices already, I think that life has been about molecular computers, building electrochemical computers, building electronic computers, which together with electrochemical computers will build new molecular computers, which will build new electronic computers, and so forth. And if you buy all of this and you think life is about computation as I do, then you look at big questions through the eyes of a computer scientist. So one big question is, how does baby know when to stop growing? And for molecular programming, the question is, how does your cell phone know when to stop growing? Or how does a computer program know when to stop running? Or more to the point, how do you know if a program will ever stop? There are other questions like this too. One of them is Craig Venter's question. Turns out I think he's actually a computer scientist. He asked, how big is the minimal genome that'll give me a functioning microorganism? How few genes can I use? This is exactly analogous to the question, what's the smallest program I can write that'll act exactly like Microsoft Word? And just as he's writing, bacteria that it will be smaller, he's writing genomes that'll work, we could write smaller programs that would do what Microsoft Word does. But for molecular programming, our question is, how many molecules do we need to put in that seed to get a cell phone? What's the smallest number we can get away with? Now, these are big questions in computer science. These are all complexity questions. And computer science tells us that these are very hard questions. Almost many of them are impossible. But for some tasks, we can start to answer them. So I'm going to start asking those questions for the DNA structures I'm going to talk about next. So this is normal DNA. What you think of it as normal DNA. It's double-stranded. It's a double helix. It has the A's, T's, C's, and G's that pair to hold the strands together. And I'm going to draw it like this sometimes, just so I don't scare you. We want to look at individual strands and not think about the double helix. When we synthesize it, it comes single-stranded. So we can take the blue strand in one tube and make an orange strand in the other tube. And they're floppy when they're single-stranded. You mix them together, and they make a rigid double helix. Now, for the last 25 years, Ned Seaman and a bunch of his descendants have worked very hard to make beautiful three-dimensional structures using this kind of reaction of DNA strands coming together, but a lot of their approaches, though elegant, take a long time. They can take a couple of years or can be difficult to design. So I came up with a new method a couple of years ago I called DNA origami that's so easy you could do it at home in your kitchen and design the stuff on a laptop. But to do it, you need a long single strand of DNA, which is technically very difficult to get. So you can go to a natural source. You can look in this computer-fabricated artifact, and he's got a double-stranded genome that's no good. You look in his intestines, there are billions of bacteria. They're no good either, double-strand again. But inside them, they're infected with a virus that has a nice, long, single-stranded genome that we can fold like a piece of paper. And here's how we do it. This is part of that genome. We add a bunch of short synthetic DNAs that I call staples. Each one has a left half that binds the long strand in one place, and a right half that binds it in a different place, and brings the long strand together like this. The net action of many of these on that long strand is to fold it into something like a rectangle. Now, we can't actually take a movie of this process, but Sean Douglas at Harvard has made a nice visualization for us that begins with a long strand and has some short strands in it. And what happens is that we mix these strands together, we heat them up, we've got a little bit of salt, heat them up to almost boiling and cool them down. And as we cool them down, the short strands bind the long strands and start to form structure. And you can see a little bit of double helix forming there. When you look at DNA origami, you can see that what it really is, even though you think it's complicated, is a bunch of double helices that are parallel to each other. And they're held together by places where short strands go along one helix and then jump to another one. So there's a strand that goes like this, goes along one helix and binds, it jumps to another helix and comes back that holds the long strand like this. Now, to show that we could make any shape or pattern we wanted, I tried to make this shape. I wanted to fold DNA into something that goes up over the eye, down the nose, up the nose, around the forehead, back down, and end in a little loop like this. And so I thought this could work, anything could work, so I had a computer program design the short staples to do this. I ordered them, they came by FedEx. I mixed them up, heated them, cooled them down, and I got 50 billion little smiley faces floating around in a single drop of water. And each one of these is just 1,000th. They're 1,000th the width of a human hair, okay? So they're all floating around in solution. And to look at them, you have to get them on a surface where they stick. So you pour them out onto a surface, and they start to stick to that surface. And we take a picture using an atomic force microscope that's got a needle, like a record needle, that goes back and forth over the surface, bumps up and down, and feels the height of the first surface. It feels the DNA origami. There's the atomic force microscope working. And you can see that the landing's a little rough when you zoom in. They've got, they have weak jaws that flip over their head, and some of their noses get punched out, but it's pretty good. You can zoom in and even see the extra little loop as little nano goatee. Now, what's great about this is anybody can do this. And so I got this in the mail about a year after I did this, unsolicited. Anyone know what this is? What is it? It's China, right? So what happened is a graduate student in China, Lulu Xian, did a great job. She wrote all her own software to design and build this DNA origami, a beautiful rendition of China, which even has Taiwan, and you can see that it's sort of on the world's shortest leash, right? So this works really well. And you can make patterns as well as shapes, okay? And you can make a map of the Americas and spell DNA with DNA. And what's really neat about it, well, actually this all looks like nano artwork, but it turns out that nano artwork is just what you need to make nano circuits. So you can put circuit components on the staples, like a light bulb and a light switch, let the thing assemble, and you'll get some kind of a circuit. And then you can maybe wash the DNA away and have the circuit left over. So This is what some colleagues of mine at Caltech did. They took a DNA origami, organized some carbon nanotubes, made a little switch, you see here, wired it up, tested it, and showed that it is indeed a switch. Now, this is just a single switch, and you need 500 million for a computer, so we have a long way to go, but this is very promising because the origami can organize parts just 1/10 the size of those in a normal computer, so it's very promising for making small computers. Now, I want to get back to that compiler. The DNA origami is a proof that that compiler actually works. So you start with something in the computer, you get a high-level description of the computer program, a high-level description of the origami. You can compile it to molecules, send it to a synthesizer, and it actually works. And it turns out that a company has made a nice program that's much better than my code, which is kind of ugly, and will allow us to do this in a nice visual computer-aided design way. So now you can say, all right, why isn't DNA origami the end of the story? You have your molecular compiler, you can do whatever you want. The fact is that it does not scale. So if you want to build a human from DNA origami, the problem is you need a long strand that's 10 trillion bases long. That's 3 light years worth of DNA. So we're not going to do this. We're going to turn to another technology called algorithmic self-assembly of tiles. It was started by Eric Winfrey. And what it does is it has tiles that are 1/100th the size of a DNA origami. You zoom in, they're just 4 DNA strands, and they have little single-stranded bits on them that can bind to other tiles if they match. And we'd like to draw these tiles as little squares. And if you look at their sticky ends, these little DNA bits, you can see that they actually form a checkerboard pattern. So these tiles would make a complicated self-assembling checkerboard. And the point of this, if you didn't catch that, is that tiles are a kind of molecular program, and they can output patterns. And a really amazing part of this is that any computer program can be translated into one of these tile programs, specifically counting. So you can come up with a set of tiles that when they come together form a little binary counter rather than a checkerboard. So you can read off binary numbers 5, 6, and 7. And in order to get these kind of computations started right, you could need some kind of input, a kind of seed. You could use DNA origami for that. You can encode the number 32 in the right-hand side of a DNA origami, and when you add those tiles that count, they will start to count. They'll read that 32, and they'll stop at 32. So what we've done is we've figured out a way to have a molecular program know when to stop going. It knows when to stop growing because it can count. It knows how big it is. So that answers that sort of first question I was talking about. It doesn't tell us how babies do it, however. So now we can use this counting to try and get at much bigger things than DNA origami could otherwise. Here's the DNA origami, and what we can do is we can write 32 on both edges of the DNA origami, and we can now use our watering can and water with tiles, and we can start growing tiles off of that and create a square. The counters serve as a template to fill in a square in the middle of this thing. So what we've done is we've succeeded in making something much bigger than a DNA origami by combining DNA origami with tiles. And the neat thing about it is that it's also reprogrammable. You can just change a couple of the DNA strands in this binary representation, and you'll get 96 rather than 32. And if you do that, the origami is the same size, but the resulting square that you get is 3 times bigger. So this sort of recapitulates what I was telling you about development. You have a very sensitive computer program where small changes, single tiny little mutations, can take something that made 1 size square and make something very much bigger. Now, this, using counting to compute and build these kinds of things by this kind of developmental process, is something that also has bearing on Craig Venter's question. So you can ask how many DNA strands are required to build a square of a given size. If we wanted to make a square of size 10, 100, or 1,000, if we use DNA origami alone, we would require a number of DNA strands that's the square of the size of that square. So we need 100, 10,000, or 1,000,000 DNA strands. That's really not affordable. But if we use a little computation, we use origami plus some tiles that count, then we can get away with using 100, 200, or 300 DNA strands. And so we can exponentially reduce the number of DNA strands we use if we use counting when we use a little bit of computation. And so computation is some very powerful way to reduce the number of molecules you need to build something, to reduce the size of the genome that you're building. And finally, I'm going to get back to that sort of crazy idea about computers building computers. If you look at the square that you build with the origami and some counters growing off it, the pattern that it has is exactly the pattern that you need to make a memory. So if you affix some wires and switches to those tiles rather than to the staple strands, you affix them to the tiles, then they'll self-assemble the somewhat complicated circuits, the demultiplexer circuits you need to address this memory. So you can actually make a complicated circuit using a little bit of computation. It's a molecular computer building an electronic computer. Now you'll ask me, how far have we gotten down this path? Experimentally, this is what we've done in the last year. Here's a DNA origami rectangle, and here's some tiles growing from it, and you can see how they count. 1, 2, 3, 4, 5, 6, 9, 10, 11, 12, 17. So it's got some errors, but at least it counts up. So it turns out we actually had this idea 9 years ago, and that's about the time constant for how long it takes to do these kinds of things. So I think we've made a lot of progress. We've got ideas about how to fix these errors, and I think in the next 5 or 10 years we'll make the kind of squares that I described, and maybe even get to some of those self-assembled circuits. So now, what do I want to take you to take away from this talk? I want you to remember that to create life's very diverse and complex forms, life uses computation to do that. And the computations that it uses, they're molecular computations. And in order to understand this and get a better handle on it, as Feynman said, you know, we need to build something to understand it. And so we are going to use molecules and refashion this thing, rebuild everything from the bottom up, using DNA in ways that nature never intended, using DNA origami and DNA origami to seed this algorithmic self-assembly. You know, so this is all very cool. But what I'd like you to take from the talk, hopefully from some of those big questions, is that this molecular programming isn't just about making gadgets. It's not just making self-assembled cell phones and circuits. What it's really about is taking computer science and looking at big questions in a new light, asking new versions of those big questions, and trying to understand how biology can make such amazing things. Thank you.
So as you hear this talk, that's the kind of a basic how people are making this kind of a two-dimensional shape. So that example was kind of pretty interesting. He can just even making those kind of a computational kind of application using this kind of two-dimensional DNAs, and then as those kind of a lecture said, using those kind of very long DNAs with a couple of the very short single-stranded DNAs, people can make this kind of a structure. And using this kind of DNAs, we can just make those kind of grow nanowires kind of positioned in specific kind of location. Like if we have a 2 electrode, which is kind of a little bit bigger than nanometer, kind of 100 nanometer also, we can just put those kind of DNA, single-stranded DNA onto the electrode, and then the other complementary DNA, which kind of a basis, configuration of the bases exactly match with this kind of a single-stranded DNAs kind of aligned on the electrode. We can just positioning this kind of a nano load structures, nanowire structures, just precisely between this kind of a 2 electrode structure. So that's the one of the idea. And using this kind of a DNA origami, we can build 3-dimensional structures as well. So the basic ideas to making this kind of a 3-dimensional structures using the DNA is basically the same as this kind of strategies. So yeah, this is the kind of toys kind of kids can play with. And in this kind of example, all this kind of yellow, green, red, and blue bar, we can make those kind of bundles of the bars using those kind of DNAs. So to create these kind of 3-dimensional structures, what people are doing is this is an actual pictures of those kind of structures created by the DNA. So people just make this kind of a DNA like this, and then using those kind of staplers, people can make this kind of load of the DNAs to be a one three-dimensional structures. And then as you can see, those kind of a short one single-stranded DNA works as an stapler in this application as well. So using that way, we can make these kind of three-dimensional cube structures. And like an Lego, we can just make multiple different kind of structures. Like in this kind of a case, we have empty holes, empty tunnels into this kind of load structures, and then we can make much more kind of a complex structures as we want. And then this is the basic design to create these kind of structures, and then all these pictures are the electron microscope images of the DNA made out of this kind of a design. And then as you can see, all this kind of observation result shows DNA can precisely make these kind of three-dimensional structures by that engineering of those kind of bases. So as I showed you in the kind of short videos, people can create these kind of structures as well. And basic is people started with this kind of two-dimensional structures, and then using those kind of ankles, you can just make this kind of three-dimensional structures as well. And this is another kind of an application. So people can make much bigger bar structures, and then based on those kind of engineering of the twist of the DNAs, people can just make the bar like these kind of twisted kind of structures. And then using this kind of engineering, people even can make this kind of a gear-like structure. So for this example, to have this is a 7 base pair case. So in this case of that, it does not have this kind of just no twist. And then if this one is smaller than 7 base pair, is kind of twisted like this, and then more than 7 base pair, it has forces like this. So using this kind of an engineering of the base pair, people can make this kind of a load of the structures, group of the DNA bundles to be kind of a curved and then twisted, and those kind of three-dimensional engineering of the structures are possible. So based on that, people can make this kind of a flat bar structure, and then this kind of a twisted bar structure, and then this is an opposite kind of a direction of the twist. And then these are all the image of the electron microscope. So using this kind of engineering, people actually can make this kind of a twisted DNA bundles. And then not only twist the bundles, people can bend this kind of bars, and then the degree of the bending is also precisely can controlled from 0 to 180 degrees. And using all these kind of options, people can even make much complex structures like a gear, and then these kind of structures. So using that way, we have thought about to making this kind of a nanoscale gear structures. We only talked about kind of a top-down approaches, which using lithography and etching and then modification and then deposition. But actually for the bottom-up approaches, we only need to use material synthesize and then those kind of self-assembly of those kind of building blocks. So these approaches using this kind of DNA origami is all used by the bottom-up approaches. And then using this way, we can make this kind of small kind of three-dimensional structures, which is a nanoscale precisely. So this is the kind of software people can design all this kind of DNA origami, and then here is the website. This is the kind of a free software. So students who are interested in kind of exploring further about this kind of designing DNA origami may find more detailed information in this kind of a website. So today we talked about the molecular self-assembly. So we talked about what is the molecular self-assembly, and then we talked about the example of the molecular self-assembly. And we also talked about the forces and then ligand materials which can making this kind of a specific molecular self-assembly process. And the DNA is another kind of a unique materials to making this kind of a three-dimensional structures, and then those kind of a design capability to creating not only two-dimensional or three-dimensional structures. This kind of a DNA origami is another kind of a very powerful method to making those kind of a small structures with a very precise control. So next lecture, we're gonna talk more about this kind of molecular self-assembly for the pattern generation in the nanotechnology. So we're gonna talk about the block polymer lithography and then nanosphere lithography for the next lecture. Okay, thank you.